Good morning. Okay, there we go. So I'm going to talk to you a bit about the new kid on the block. I'm going to take you through the journey of how we actually discovered the virus on macadamia. So in 2019, myself and the people who participated in this project, David Reed, Genevieve Thompson, and Nicola Roberts, started all bright-eyed and bushy-tailed that we're going to solve the macadamia chlorosis disease. Because we were certain for a fact that this is actually caused by either a virus or a fastidious bacteria. So what we did is we came down to the Mpumalanga Lowveld and we visited 5 farms as well as one commercial nursery. And then we moved on with the slide and we took samples ranging from different ages, from nursery trees up to more mature trees of about 25 years in age. We also included different cultivars ranging from Nelmax, Beaumont, so all of your major cultivars were also included. And then we noted the different disease symptoms which we observed, mainly focusing on the chlorosis that we are observing. And we also included healthy trees so that we had something to compare our data with. We also noted trees that had some stunting, dieback and necrosis and mottle as well. And these are just some of the symptoms that we looked at. So obviously, the chlorosis is the main symptom that we looked for. We also noticed some necrosis and some mottling as well. So we then took the samples back to the lab where we extracted specifically RNA. So when you look at your total nucleic acid composition, I'm sure a lot of you have heard about DNA already. So we specifically focused on the RNA, which is a more minor component of your genetic material. However, this filters down for the viruses that we were trying to hunt, as well as your actively replicating bacteria. So then we compared the data sets between the chlorotic trees and the healthy trees. And there all of everything stopped. We couldn't find a clear difference between the chlorotic trees and the healthy trees. So a little bit less bright-eyed and bushy-tailed at that point. However, we were able to identify viral reads within 5 nursery tree samples. And these are actually the nursery tree samples which we saw. And the symptoms that we noted here were some chlorosis. You got stunting as well, and this rugosity that basically looks like a shoestring that's been pulling up the leaf. So we decided, okay, as virologists, we're going to focus on this and see where we get with that. So we took the data that we had and we looked at the entire genome component of the virus, and we were able to identify a member within the orthotospovirus genus. So just some further characterization, so the main marker for this group of viruses is the nucleocapsid protein, and we compared this to the known viruses within this group, and we actually found that this is a unique virus within this group. And just, I'm quickly going to point this out here, and we're getting to this a bit later on again, is that there are 5 phylogenetic clades or groups for this virus, but as I said, we'll get to that a bit later on. So what we know about orthotospoviruses is that they cause this concentric ring spot symptom on the plants which they infect. And we know that we actually saw this when we were in the orchards. So we decided to maybe try and explore this to see whether or not there's a correlation between this ring spot symptom that we're seeing out in the orchard and our virus. So we optimized the virus-specific test for this virus, for the detection, and came back and did some sampling in 2020, once again focusing mainly on the Mpumalanga Lowveld. And then in 2021, we got samples from Mpumalanga, the southern coast of KwaZulu-Natal, as well as Limpopo. Again, we took trees in from different ages, ranging down from nursery trees up to more mature trees of about 25 years in age. Again, looking at different cultivars. And here I just want to emphasize that we specifically looked for that concentric ring spot symptom. So all of the samples that we tested actually tested positive for this novel virus. So to further confirm this association with the ring spot symptom, we just did a little pilot study in which we took leaves from a single tree showing different symptoms. And the symptoms we focused on, I'm sorry, I'm just going to focus on that screen with the laser pointer. Else we're going to start doing a little dance. So we had the ring spot symptom. Here is some mottling that you can see there. That's not as clear on the picture, but it's a little fine little specks that you see on there. Some necrosis as well, and then some healthy controls. And only the ring spot symptom tested positive for this virus, further confirming that the virus is actually associated with this ring spot symptom. So due to the association of the virus with this ring spot symptom, we propose to actually call this virus macadamia ring spot associated virus. It's a nice thing about virology, if we can give things English names. We don't have to go back to our Latin. We identified it also, which is a bit of a concern, from all of the major cultivars which are being planted. And once again, this is limited to the samples which we actually received. We do not know yet, maybe all of the cultivars are susceptible, but we do know that these cultivars are susceptible to the virus. We also identified it from the 3 largest production areas, so Mpumalanga, Limpopo, and KwaZulu-Natal. And once again, this is only because due to the lack that we didn't actually get samples from the other production areas. So if you are sitting here and you have actually seen the symptom in your orchard, please submit it to our lab. So I'm with Marita. A lot of you know Marita Skuman at the ARC. I'm usually around the office. So if you can please submit me some samples. So I just also want to caution that this is actually not the first time that a virus has been identified from macadamia. So there is a report that's all written in Chinese, but if you use your best Google Translate skills, you can actually get to the crux of what's going on in the article. And they found a virus that actually belongs to the same group as the virus which we identified our virus from, so the orthotospovirus genus. However, the symptom expression that is cited within this article is different to our ring spot symptom, which we found for our virus. And they also identified the virus as a known virus, or a virus that has already been described and is known as watermelon silver mottle virus. And the symptoms associated with the virus in China is actually this chlorotic symptom with some necrosis as well. I went back to some of the viral reads that we generated for the data that we had, and I could actually not find this specific virus in our data reads. So why should we actually bother looking further at this virus? So it comes back to the group in which it falls, the orthotospovirus genus. So this is a group of viruses of about 25 species, which are known to infect over 1,000 plant species, ranging over 90 botanical families. So we do know that these viruses are transmitted in a persistent manner by thrips. So this just means that should a thrip actually acquire the virus, it will remain infective throughout its entire lifespan. So as I also previously mentioned, we do have 5 distinct groups within this clade, and members within each clade actually share the geographical origin as well as the thrip species which are responsible for vectoring these viruses. However, as everything in life, there's an exception to the rule. So the group in which our virus falls, the 2 known species within this is groundnut yellow spot virus as well as groundnut chlorotic fan spot virus. So both of these actually originate from different geographical origins as well. And now with the first virus within the orthotospovirus being described from Africa, ours is also quite unique in that regard. But one thing that these 2 viruses do have in common is that they are vectored by skirothrips. I really apologize, Skalk, if I'm pronouncing that incorrectly, dorsalis. But from the research and the literature that I've looked at is that this is actually not a thrip that's occurring on macadamia in South Africa. We do, however, have skirothrips, the citrus thrip, that is very common on macadamia especially the Mpumalanga lowveld. And therefore, it is plausible that this might actually be the vector which is responsible for transmitting this virus. However, we do need to do further research because there are a number of thrip species that do occur on macadamia, and any of these can be candidates for this to vector this virus. So what is actually of more concern? The type species for this group is tomato spotted wilt virus. So if you've ever farmed with tomato in your life before, you'll be quite well known with a disease known as Kromnak disease. And Kromnak is associated with tomato spotted wilt virus. And this virus is still considered to be one of the most destructive viruses globally, or contributing to billions of losses in income annually. So what are we going to do about it next? So that because of these losses that can be associated with members within this viral group, we have started a project. It's a collaboration between myself, Marita Skuman, and Liesl Mori, who's just our statistician at head office. where we want to determine the impact of macadamia ring spot virus on yield. So this study will be conducted over 3 years, where we will compare yield data from trees showing the viral symptoms with trees that are not showing the viral symptoms. And we also want to actually monitor the spread of the virus within the single tree canopy and to the neighboring trees as well, to see how efficiently the virus is spread. What we also want to see is whether or not macadamia ring spot virus actually predisposes trees to secondary infections. Because as with humans, if you get a viral infection, your immune system is compromised, and therefore you get sicker easily by secondary infections. The same goes for trees. We also want to determine the viral incidence in nurseries to see how common the virus actually occurs in your nursery trees. Because again, as with humans, the younger the tree, the more susceptible it will be to the virus. And we also need to determine whether or not there are other symptoms associated with this virus when we specifically look at nursery trees. Because the initial trees from which we identified the virus were nursery trees, and all of these actually showed different symptoms to this concentric ring spot symptom with which we now associate the virus. So that is short and sweet from my side. And there's just my email address, so please, if you have any samples, you are more than welcome to submit them. Thank you.
Ronel, we're nicely ahead of time. Thanks for that. And I'm sure there's someone inside in the audience that really would like to ask you a question. Guys, are most welcome.
Lights are very bright here. Can't see them.
Maybe I can ask a question, Ronel. It's well known that your thrips can transmit viral diseases on various crops.
Yes.
Is it possible? I'm just curious. I don't know. How do they transmit it? Is it in their saliva? Can you test thrips to see if the virus is on it?
Yes, you can. So it is transmitted by the saliva. So especially with this virus, so the orthotosphoviruses are actually capable of replicating within thrips as well as in the host tree. So that's why we say it's transmitted in a persistent manner because of this replication within the host. So it is transmitted by the saliva. And we can test thrips, it's just they're very tiny. So it's a bit difficult, but we can.
Thanks, Ronel.
Okay, there's a hand.
Good morning, very interesting talk. I would just like to know, have you looked into the disease progression yet of this virus?
Not yet. So this is still a very new project. We only really characterized the virus within the last year, 2 years. So we're starting fresh now to try and get all of that sorted and see the disease progression and everything. But it's still very new, yeah.
And your feeling while working with this virus till date, the effect that it might have on yield or production?
No idea yet. That's why we're starting this project, because we need to be certain what effect it actually does have, because we're not sure yet whether or not it also infects the nuts, because we've only focused on the leaf so far. We haven't actually tested any nuts to see if the virus is in the nuts. So that's the 3-year project that we're launching now with SAMAC to try and determine this.
Okay, great. Good luck.
Thank you.
Any more questions? Oh, there's a question.
I'm not sure if you mentioned it, but does this have anything to do with the sclerosis or the yellowing of the macs?
Unfortunately not, no. No, we couldn't find a link between the virus and the chlorosis. Sorry about that. Alicia is going to be under fire with that as well tomorrow, talking about some of the data that she generated from the chlorotic trees. But yeah, we don't know yet.
Hi. Have you, or are you planning on doing any research on mineral profiles in, let's say, a leaf analysis that results in the tree being more susceptible to the virus?
For the virus, not specifically. We haven't actually thought about doing that, but that might actually be a good angle and something to think about. So thank you for that. But yeah, for the chlorotic trees, once again, I think Alicia did some work into that as well, which she'll present tomorrow.
Ronel, I think unless there are other questions, I think we can let you